Good afternoon, and welcome to this panel discussion on Immuno Oncology partnering. It is my pleasure to welcome with my co-chair, Duncan Young, an illustrious panel that will focus over the next period in all the different trends and events and what is happening and what will continue to happen in the partnering space, specifically in the immuno-oncology, such an important field. And that has some peculiarities, make it slightly different to the broad partnering, and we will focus on the differences and opportunities in that field. I am Esteban Pombo. I am CEO of TargImmune Therapeutics, a private company based in Basel, focused on targeted immuno-oncology approaches. And I would like to give the word to my co-chair, Duncan Young, for a brief introduction, and then we'll have introduction of every of the panelists. Duncan, please.
Great, thank you, Esteban, and thanks to Sachs Associates for hosting us and having this discussion. My name is Duncan Young. I work within the oncology business development team at AstraZeneca on search and evaluation, so scouting for technologies and opportunities across all stages of development to support our oncology pipeline. Our areas of focus are around tumor drivers and resistance, DNA damage response, antibody drug conjugates, and of course the topic of today, immuno-oncology, with growing interest in epigenetics and cell therapy approaches as well. And of course, I think a topic that we'll come on to touch on later, understanding how combinations between those can sort of drive patient benefit as well. So very looking forward to the discussion today and the opportunity to host, as Esteban said, this illustrious panel. So perhaps I'll pass on to Khatereh first to introduce yourself.
Sure, thank you. Hi everyone, I'm Khatereh Ahmadi, and I'm part of the global business development team at MSD, which operates as Merck & Co. in North America. And I'm responsible for oncology business development for European opportunities. So I've been with MSD for about 5 years, and prior to that, I was very much at the startup end of the business in various CEO and business development roles. And prior to that, I started my career as an academic in the cancer biology area. So just a couple of words about MSD in general. So we're headquartered in New Jersey with 4 business areas, which is pharmaceuticals, biologics, vaccines, and animal health. I'd say that we're active in all major therapeutic areas, and oncology, including immuno-oncology, is one of the major pillars of growth, with Khatereh being our foundational program.
Thank you, Khatereh. And Aram, would you like to introduce yourself?
Sure. Thanks, Duncan. My name is Aram Mangasarian. I'm the CEO of Noxon Pharma, which is based in Berlin, Germany. We're developing therapeutics to target the tumor microenvironment, particularly via chemokines, which turn out to be quite important in the movement of various cell types around and within tumors. So our lead asset's called NOX812, and this has effects both on the exclusion of effector cells and also the repair of damaged components of tumors by vasculogenesis. And so I think of interest here is the fact that we're developing this in multiple ways. First, in combination with immuno-oncology agents like PD-1 inhibitors. We've collaborated with MSD in that, but also in combination with radiotherapy because of its effect on creating hypoxia and vessel destruction. So those are the things we have to think about as we think about partnering and the business development long term. Thanks.
Thank you, Aram. Carlos Buesa, we've got 2 Carloses on the panel. Carlos Buesa, would you like to go first?
Buesa, that's me.
Yes.
Okay, thank you. Sorry for that. So good morning, everybody. I'm really glad to be here with this illustrious panel, as Esteban said. So I'm Carlos. I'm the founder, I'm CEO of Oryzon Genomics, a company that we started back in 2000 as a functional genomic and biomarker discovery and functional genomics company. I am a molecular biologist by training, and we have become over time, I would say, the European epigenetic company, so to speak. And we are mainly specialized in one of the most intriguing targets in the area, which is LSD1 and histone demethylase. The first histone demethylase that was characterized at the time. So LSD1 is basically having an important role in a number of important physiological processes through development and also in pathologies. And it plays an important role in CNS and also in oncology. So we have explored, we have become the LSD1 company on the market with 2 programs in phase 2, the one in CNS and the one in oncology. And in oncology, what we have seen in those years, I mean, we were the first moving an LSD1 inhibitor to clinic, doing a first-in-man in refractory relapse acute leukemia. And we are also exploring LSD1 in a different set of solid tumors. What we know now is that LSD1 is a fascinating target in a subset of oncological diseases where it might play actually a dual role in a cell autonomous way, basically having a direct action. And basically this is clearly shown, for instance, in small cell lung cancer, a tumor which is particularly not working, not of the best areas for immuno oncology so far. And this is basically driven by a specific transcriptional balance and reactivating notch and S expression. But on the other hand, on this particular tumor, what we have seen is that there is a cell non-autonomous effect. And that was particularly relevant. That was actually being shown by the work of Yan Shi in Harvard, where they demonstrate that LSD1 ablation stimulated anti-tumor immunity and enabled checkpoint blockade. But what we have seen is that these experiments that were done by knocking down with our LSD1 inhibitors, our pharmacological effects, basically are able to produce the same effect. So we are able to see a very strong change on the immune system of the host on our model. So we believe that there is a particular important avenue of combination with immuno-oncologic agents, particularly in the neuroendocrine tumors. So we are keen to explore that, and we are preparing a number of clinical trials that we will probably inform this year, exploring these avenues.
Great, thank you, Carlos. I might then move next to Richard.
Hello, thank you, and thank you, Sax, for the invitation to speak this afternoon. I'm Richard Godfrey, the CEO of BerGenBio. BerGenBio is dedicated to developing first-in-class inhibitors against AXL tyrosine kinase. We really are world leaders in understanding the biology of AXL and how it mediates aggressive disease, particularly sort of immune-evasive, drug-resistant and metastatic cancers. Our lead program is bemcentinib, an orally bioavailable, highly selective inhibitor of AXL, currently in multiple phase 2 programs as monotherapy, combination with chemo and checkpoint inhibitors. We're approaching the end of a number of these proof of concept studies with very encouraging results and preparing to communicate our phase 3 and registration path. In addition, we're developing an antibody against AXL. We've just announced the second part of our phase 1/2 study there. And probably the most immediate news point to mention is that AXL is also used by the COVID virus, by SARS-CoV-2, both to enter host cells and also to dampen the type 1 interferon response. We've now conducted a clinical trial in South Africa, India, and the UK in hospitalized patients taking Ben-sensitive monotherapy, and we're preparing to release top-line clinical data at the end of this month. So very excited about that. Very busy program. Hopefully, we can contribute to the fight against COVID, as well as maintain our efforts in leukemia and solid tumors. So looking forward to this conversation, and thank you for allowing me to be here.
Thank you, Richard. And it echoes some of the earlier panels and the discussions about how fascinating it's been to see biotech and pharma rise to the challenge of COVID over the last 18 months. So thank you. Finally, then, Carlos, please.
Yes, the other Carlos, yes. Thank you, Sachs, and thank you to Esteban and Duncan for the invitation. I'm the CEO of Ultimovacs. Ultimovacs is a public company located in Oslo, Norway. And we have a new generation of universal cancer vaccine that is the off-the-shelf product that we are currently in phase 2 with approximately going to enroll 500 patients in multiple indications: melanoma, mesothelioma, ovarian cancer, and head and neck cancer. Also in combination with multiple checkpoint inhibitors like Pembro, Ipi/Nivo, but also Durvalumab and Olaparib in the ovarian cancer. And we have collaborations with BMS and with AstraZeneca for these studies, and groups, specialized groups in US, Europe, and Australia. And the mechanism of action, basically, we enable and train CD4 cells to recognize telomerase, cells expressing telomerase. What is, 85 to 90% of all cancer cells, regardless of the type of tumor, express telomerase. So it's a very good target, and we already have quite good phase one data in long-term survival, and we expect to also be able to present at a medical conference this year very exciting data in combination with Pembro in melanoma. We have a second platform that we just initiated phase 1 trials. So very happy to be here and give my perspective from both the big pharma side, where I was in multiple companies, but also now for the biotech.
Great, Carlos. Thanks very much for this. We have the broad view of the panel, and one of the questions that has been asked multiple times is really: is the world starting to get tired of immuno-oncology, or is immuno-oncology just at the beginning of a really very important potential going forward? And I think what we will focus on is really the deal making. There are other panels that will focus on innovation, but we cannot separate deal making from innovation entirely. And it's also very important that we have learned many things over the last year. Richard mentioned that the opportunity in COVID emerged due to an insight into the biology. And COVID as a virus clearly has something to do with immune stimulation, and the immune response to cancer is what we're trying to modify. So with this really picture in place, I think we can focus on what have been the effects and the learnings, both for deal making and biology and overlap, in the last 12 months, where we have experienced this lockdown pandemic. We have learned new ways of communicating, new ways of dealing with the way that we approach a biological problem and convert it into a therapy, and then how do we work together to take that therapy to patients in combination or as a single drug, and of course collaborating with pharma and biotech. So that is really the big picture of what we want to address. What have we learned, and what is changing? What are the trends going forward in deal making? So, Duncan.
Thank you, Esteban. And you said we're conscious that we're treading a line between some of the other panels talking about the innovation side. But if you look back at some of the deals that have happened over the last 12 months during the pandemic, again, you're really reflecting some of the way that the IO landscape is broadening beyond the original checkpoint inhibitors. We're seeing bispecifics, we're seeing myeloid biology coming to the fore. We're seeing modulators of the tumor microenvironment been more of interest. How cytokines, cancer vaccines, cell therapy fits into this is something that clearly companies are all trying to assess, and what the broader IO landscape will be, both with those as monotherapy approaches, but also then understanding how you can drive combinations to get efficacy in patients that are non-responders. So we've seen heavy deal activity over the last 12 months. And it doesn't really seem that the pandemic itself has had too much of an impact on that. But perhaps as we're hearing the discussions, the question is how much of that was ongoing relationships, and how are the new relationships being forged during the lockdown. So on that point, Khatereh, it'd be good to get your perspective from Merck on how you've seen the pandemic impact your deal flow and relationship management during the lockdown.
Yeah, sure. So, I mean, we did actually some stats on this in the BD group. So, 2020 was a very busy year for our business development team, and it was a very productive year. Actually, kind of stood out amongst the previous year or 2 years. Which is, we didn't know what to expect, so that was nice. We signed a number of deals, and some of those in the oncology space, some big deals that had to be done very quickly, like the deal we did, for example, with Velos Bio, and all of that was, of course, done online. And I personally was part of some deal negotiations, complex diligent processes, and I must say, all of that, worked pretty well. Obviously missing the face-to-face, but I can't say that it impeded or slowed down our process in any way. I mean, it remains to be seen what will happen this year. In terms of, outreach and search and evaluation, I'm more proactive at doing that, because obviously we don't have the face-to-face conferences. I actually reaching out to VCs and my contacts to make sure, that there is a pipeline, because we don't have that, kind of face-to-face time and serendipity kind of interactions that we used to. And so, it'd be interesting to see what happens this year. But certainly the past year, I would say, was better than normal.
So, Khatereh, from that perspective, it might be that we are actually wasting less time traveling and doing more time relaxing in our pleasant environment and actually getting work done, rather than wasting times in hotels and airplanes. But on the other hand, you mentioned this loss of face-to-face interaction, the ability to bump into people across and so on, what thing that we are missing. And I believe there some aspects that we really need to rephrase. So, for example, Carlos, to say Carlos de Sousa, not Carlos. Carlos, if I may ask you, Carlos, you are as a medical doctor, and looking at the opportunity to interact, are we missing on the ASCOs and the ESMOs and these medical conferences? Does that have an impact?
I think that we see some impact. Starting, of course, with the ASCO last year. During these conferences, in addition to, for instance, the partnering activities, because, the large pharmas have their full teams participating in this conference, so it's easy to meet everybody in the company that's involved in that project. But on the medical side, yes, now we can attend and listen to presentations, but the part where we are missing a lot is the, advisory boards. Normally, the team will plan a lot of investigative meetings. What we could do in one session, now we need to do in multiple. Have to do in multiple sessions because country by country, what increases the number of activity. And yes, we miss a lot of that interaction with physicians. Discussing projects, the feedback, what is new? As you know, a lot of these conferences, we get a lot of information, things that are not necessarily being presented in posters or oral presentations, but we know what is happening in the field, and we miss that part. And yes, we know it's, advisory boards and meetings with the investigators are much harder.
So this serendipity that often leads to connection with other connections that is being lost. On the other hand, there as Khatereh mentioned, you can have a structured approach to really be proactive in a different way. And I was wondering, Aram, if your experience in this year in terms of interacting with investors or other elements that are of course important in enabling you to structure a deal or have opportunities going forward, has that been different for you?
Yeah, yes, it has. I mean, I've had to rely much more on my board and some other external advisors to initiate transactions or to initiate discussions, I should say, because without the ability to turn up at the conferences and go to some of these meetings with full teams, it's quite difficult unless you have an established relationship. And when we were sort of quiet for a couple of years as we were restructuring and kind of replanning the programs and getting things moving again. And so, a lot of those relationships sort of got stale. And so, yeah, now that we have a lot of momentum again, we have to rebuild those. So that's been, and I can actually, Khatereh, with MSD, it was interesting, the way that our collaboration got going. It was at one of the conferences where we had one of these typical meetings where we walked into a room with, 20 people from MSD. And, I think it was Eric Rubin who liked the program and said, yeah, that's interesting. And we should try and do something together. So that's the kind of interaction, like Carlos mentioned, that you miss and is much more difficult to get now.
Are there some interactions that are easier, given that everybody is in their home and maybe have time for you that otherwise may not have had time for you before?
Definitely. On the investor side, particularly, that's gotten a lot easier for us as a European-based company that's not in one of the big financial centers. I feel like we're on a much more equal footing with the other companies because, it's just a video call now. And the fact that we're far away doesn't really make a difference.
I second that, actually. I mean, it's much easier. I don't know how you find it, Duncan. It's much easier to schedule meetings with some of our key scientists than it was before.
Yes. Yeah, no, I completely agree. The only caveat I'd had is that I think we're now coming up to the year anniversary, and there is an element, I think, of people being a bit fed up with the lockdown, fed up from working from home, and an element of burnout because people, the fact that you can never get too far away from your laptop because you're in lockdown means that people are dragged into calls at I think we've felt ourselves dragged towards the US time zone. They felt themselves dragged towards Europe in particular. So yeah, absolutely. I think it has the flexibility has had its real benefits, but there's a balance there that I think is wearing a little thin as well.
Yeah, I think that is going to be, as you were saying, going forward. That's the part where it's going to start to be a stretch for the teams. In terms of how long do you really want to have your calls? Because in some of these conferences, you are with China, Japan, and then you start very early, and then you are at the US. You have calls at 8:30, 9 o'clock in the evening. Is there something that you want to continue doing for a long period of time? So I think that puts a little bit of an extra burden on the team because the days become quite long.
Yeah,
I think there's possibly one other element as well, particularly for small companies.
So it has challenges and opportunities.
Sorry, hello?
So go on, Richard. I think you were
I was just going to say there's one other important parameter to remember, and that's that as a small company, being able to maintain surveillance on developments across a spectrum of therapeutics is quite difficult if you're observing through a screen, through a monitor. Whereas the ability to network and speak with people by those coincidental contacts that you make and just sort of say, what's new? Did you see this? What's happening here? Is extremely useful sort of landscape information that's sort of otherwise quite difficult to assimilate, and you can easily miss something that's very important. So that's a really valuable element of sort of the serendipity and the sort of brownie emotion that we get from attending. So I think the conclusion is going to have to be a fine balance between being efficient and working from home office and using our time wisely at conferences to get the maximum benefit from presence there.
Right, Richard, thanks. I think, changing tack a little bit, maybe we say clearly there are some opportunities and there are some challenges in the way that we do business, but it has been very productive for everybody. Really, we found ways in which we can cope with the deficiencies. Whether that would be the model for the future remains to be seen. But one important element is really in immuno-oncology specifically, have there been changes in partnering models or data requirements that have been driven by further understanding of the science or further experience, given there's these thousands of immuno-oncology combination trials, not many of them prove to be positive. How does the landscape change in terms of requirements and partnering models for pharma, and how does the biotech community probably approach pharma in order to be able to work as best as possible in this ecosystem to generate value for the patients and, of course, value for the whole community? So I don't know, Khatereh, do you have a perspective on this?
Well, I think that, the dawn of the immuno-oncology, that there was a lot, and there continues to be, a lot of kind of excitement and hope, with CTLA-4 and PD-1 and a lot of other agents that were kind of built on this. You inflame the tumor and then you come in with the checkpoint inhibitor and, bang, you're going to wipe out the tumor. And the tumor is a very complex structure. You have tumor cells, of course, you have the microenvironment, you have, stroma cells and the fibroblast, they're all playing a role. And so, some kind of, I think, some modalities and agents where we thought, mechanisms, actions where we thought were going to be easy wins, have turned out to be not fruitful, let's say so far, or we don't know how to use them. So it's become more entrenched and more complex. Although I don't mean that in a negative way, that's the way science is. And as we learn more, we're finding other ways and other agents that work. And for example, one of the learnings we have is that agents that work as single agents, have single agent activity, have more chance of working in combination with anti-PD-1. Hence our interest now in ADCs, as an example. And this wasn't the thought process in the beginning. For example, with IDO has no single agent activity, but we thought it could modulate the tumor environment in a way that didn't matter about the single agent activity, would work in combination. So I think that we are learning more about the complexity and having a more gathering a more in-depth understanding. And also immuno-oncology, I think, has obviously now is gone beyond the checkpoints and the kind of agents that we know directly modulate the anti-cancer immunity, and KRAS, for example, or other, what we used to think were tumor intrinsic targets, are now have an impact on immuno-oncology because mutations in KRAS, for example, can change the tumor microenvironment and make it more immunosuppressive. So maybe I'm not going in any particular direction, but I'm just saying that, you know, it's become more complex, but we're learning more.
I think that's
has that affected deal structure, deal terms? Yes, go ahead.
So you're asking me? Yeah, so I mean...
And that are from that perspective, does that affect the deal terms of the structuring? Yeah, go ahead, Khatereh.
Oh, sorry, okay. Well, I mean, you know, I think that it has in a way, because I think that preclinical data, which we, you know, is more difficult to predict the translation into clinical data, and that's, you know, stating, you know, maybe the obvious, but it seems to be particularly the case with immuno-oncology, or somewhat pronounced, and that has, you know, changed, I think, deal structure in a way, in that we have to kind of think about how we manage that risk in the deal structure.
And I think you're absolutely right, Khatereh. And we've seen things move on from some of the very early, very expensive bets on IO combination opportunities that happened a few years ago that didn't pan out. You touched on an IDO, but we've seen other aspects of checkpoint inhibitors that have not really proven in the clinic in combination. And so I think there is that element of caution, element of better understanding of the biology and better understanding of still some of the black box aspects of the biology, and recognizing that these are only really going to be teased out in the clinic. And that's where I think where we look at the opportunities for partnership in immuno-oncology is really understanding how can you quickly get to a point where you can get that no-go signal in the clinic and design the studies smartly to do that, hopefully driven by some understanding of the biology and the biomarkers you might do to identify the patients that are going to best respond to that combination as well. Yeah, I think, and that's got to be driven by understanding the biology of your agent, but also across the particular indication that you're going into, because we see the indication is equally a determinant as to what sort of response you'll see to the various IO and IO combinations. But yeah, I think that increased understanding of the biology around a particular agent is perhaps what you were getting at earlier, Carlos Buesa, when you were talking about LSD1 and the only by developing the agent as you have, you've been able to get these insights into the potential in IO.
No, it's indeed it's very much as you said, and I see that the small cell lung cancer might be a beautiful example to these general considerations about what agents may or not work, if they work in playing solo or in monotherapy, or what would you be expecting in combination. In our case, what we were having an interesting collaboration with our drug that was done at the Fred Hutchinson. And what we saw is that there are certain tumors or in PDX settings, so cells coming from relapsed patients, they were extraordinarily sensitive to LSD1 inhibition, whereas others, they were not that much, and others, they were absolutely not. So this understanding of what is this complexity behind this difference of responses is fundamental. So we were studying for a period of time the different biomarker profiles. We came across a set of biomarkers that we were actually using on our signal-seeking study that we did in small cell lung cancer, and we do believe that there are a number of a certain profile of patients, molecularly defined patients that might be much more or much better candidates to a specific actions. That said, there is still an open question, because in our case we have a clear activity, because as I said, there is a clear reactivation of NOXXON S1, and we see a deactivation of ASCL1 and other oncogenes driving the tumor. But it doesn't preclude that some of these patients that are not responding on monotherapy might actually respond on a combination. So our approach here is basically probably in this specific setting would make more sense to get a broad approach and to do a post-hoc analysis to see if there is a difference in the synergies between those patients that were ab initio already responding to the LSD1 monotherapy compared to the others that they were not. So I think is certainly the path to go, as you say, Duncan, the biomarker and the profiling. Otherwise, I mean, it is going to be very expensive bets constantly, and I don't think that even the big pharmas are for doing expensive bets. So we need to try to get a rational, but we need also to understand that some of these synergies may happen with or without a prior mono activity in monotherapy.
Thank you, Carlos. And that's perhaps a good segue into the next question, which is going to be around how are we seeing the changing landscape and availability of capital altering the drivers for biotechs when they're forming these partnerships, and how they look to structure them and what they get out of them. But obviously a proposition which is to go into a signal searching study in the clinic is very different to something that a pharma company can be confident and picking up and running with straight away. I'm conscious, though, before we come on to that, we have a question from Andre Brundley, which is asking about the microbiome. And I think perhaps before we move on, it's a good opportunity here to say that a lot of the discussion about needing to understand the positioning in the clinic, needing to understand the rationale for patient selection and response here, equally apply in that setting. And I think that there's still, you're introducing a lot of additional variables when you're looking at the microbiome research that we need to fully understand how we progress that. Khatereh, quickly, I don't know if you've got any other thoughts on.
I mean, certainly it's an important area and an area of interest. In fact, in one of our discovery centers, we have 3 in San Francisco, Boston, and UK. And in Boston, we have quite a big effort on the microbiome. But I think it's still a bit of a way to go before we really understand how therapeutics can be positioned in the clinic, how they work. But yeah.
So Esteban, should we turn the question to the biotechs?
Yeah. I think let's. What I was thinking about when Carlos was talking about the importance of generating understanding at the biological level, and Richard said, well, we are experts in understanding this particular pathway, and Aram with his view of this chemokine and how this works. That's all really fundamental biological understanding. But then the question is, how does that? What would be the partnering strategy around that? Basically, on the one hand, you said, well, let's work with a pharma partner that has then the breadth of capabilities, so we can look at this LSD1 not in one indication, but let's look at 100 cancer indications at the same time, just to give a number, right? Which is beyond what a single biotech company can do, unless it's a very broad opportunity. So how does that drive a potential collaboration with pharma? First of all, from the biotech perspective, and then second from the pharma perspective. And then we can also look at that in the context of the availability of capital that you said, right? A biotech company may want to do everything if you have enough money to do so, to build capabilities, but sometimes it's much more efficient to work in collaboration with somebody that already has established infrastructure. So who would like to take that point?
Well, okay, yes. The example that you mentioned, analysis one, I think in our case, what we have seen is that there are a number of solid tumors where this might make sense. Small cell lung cancer is certainly one of them, but we have seen that there is sufficient emerging evidence to support a case for in general for neuroendocrine tumors. So where these neuroendocrine tumors are basically sharing some core features with the small cell lung cancer, but there are also some differences which are quite important. So I think that's for instance this concept of a basket trial, signal seeking with the idea to be able to enrich a certain arm to make it gain registrational merits is something that is certainly having a clear interest for the biotech point of view, and I tend to think that might also resonate on the pharma companies as well, because this is basically sort of defining expedition that might narrow the effort that you want to make down the road to make a regulatory path.
You know what we are doing,
and it will be data-driven based on a good study. Yes, Carlos.
No, I was saying, it depends on the type of molecule that you have. We are at the moment well funded, so that's why we can run four phase 2 studies with 500 patients. But the question is, what are we going to do afterwards? Because basically, we can be used across, a big quantity of solid or semi-solid tumors. We are doing at the moment in 4. We have multiple combinations. That's the goal, that we want to get all this information. We are making big investments in CMC, and we can do that because at the moment there is capital available. But then the big question afterwards, as you are saying, a biotech, can run phase 3, maybe 1 or 2 indications, but then we will be leaving a lot of value on the table, because in reality, from what we expect data and possibilities, we could be, or a strategic partner could be going after 5, 6, 10 different types of tumors using our universal cancer vaccine. That's the part where I see that we can have the cash that takes us definitely until the phase 3, and then is more, how much do we want to finance the company for a limited program versus a partnership to go with a broad and increasing the potential of the product later on in the market.
Yeah, I totally agree.
Richard or Aram, please.
I was just I agree with Carlos that it's difficult when you've got multiple options with your compound, and with us it's for example with the PD-1 combinations, but also radiotherapy combinations, and where do you park an asset like that where you have multiple different approaches you'd like to pursue? And so, we've had to change our view a bit about how we think about it. We're starting to look at it just simply trying to do risk-sharing, non-licensing type transactions where we say, all right, listen, we both have an interest. Where's the overlap in interest? With the PD-1s, we'd argue it's in these solid tumors where you don't have activity because you can't get the effector cells where they need to be. That's where our drug can help. And by the way, for some of the cell-based therapies, we're getting similar types of discussions restarting now with some companies, which is interesting because a few years ago they didn't want to talk. So we've had to change our thinking quite a bit in terms of how we want to look forward and develop it. And certainly, as Carlos said, our ability to finance or co-finance these downstream is very important for that strategy to work long term.
And we're coming into the final stretch, but Richard, did you want to pick up on your comment there?
Well, I was going to say, everything that's been said, I agree with. Our strategy, of course, has been to demonstrate proof of concept in several different spaces, but at the same time, trying to keep an eye on where the puck is going, so that when we ultimately get to defining a first path to registration, it will really be into a space where there's an unmet medical need. In our case, it's in relapse AML, which has emerged after the innovation in the first line AML. So again, using our technology and our surveillance to find a path forward, but without doubt, there are much larger opportunities that many of the technologies presented here could pursue, not least of all AXL inhibition in solid tumors in combination with checkpoint inhibitors, chemo, etc. And then the last thing, of course, is I completely subscribe to the idea that a therapeutic that has monotherapy activity certainly is most likely to work in combination with others, particularly when you're promoting the immune system, and even more valuable if you can identify the patients with biomarkers. So I think it's about sort of sharpening your focus so that you can actually get something over the line, as well as represent a huge potential should a pharma partner want to be with us. But we have to be responsible for developing our own assets, our own technology, and serving a patient need. I think everything else will click into place. And I say we are in a fortunate economic environment now, so that capital seems to be available, so long as we really are adding value to the patients and addressing those medical needs.
Absolutely.
And as you say, well, the importance of being able to focus on an asset, understand the biology, and go deep in the biology, and be open to opportunities like Carlos in Oryzon, and then suddenly we find that there's an opportunity there, or in the chemokine space, or that. So clearly, for Duncan and Khatereh, having a very strong biological rationale plus biological data, it makes it much easier to find ways in which work together. Patient selection, the biomarkers, the design of the clinical trials, also extremely important. So that takes us really pretty much to the end of our panel discussion today. Really, there are a couple of themes that we touched on that I believe are very important and are here to stay. One of them is that we learned new ways of working last year, and some of these new ways of working will continue to be in effect even when life returns to normal. Really, there's opportunities to exchange information in a virtual setting and learning how to use that most efficiently. But then really taking patients into consideration, how to bring therapies to patients, the model of partnering is still there and the opportunities for going forward. So with that, thank you very much to all our panelists and my co-chair, Duncan, for all the work and collaboration you've done today. Thank you very much.